If you celebrate Thanksgiving, I hope you enjoyed a great Thanksgiving and at least that somewhat long weekend. At times like this, time of Thanksgiving, I'm reminded personally of how grateful I am for the truly extraordinary colleagues that we have here at Brown. It's really amazing. I mean, sort of seminars like this, workshops, having conversations, informal conversations with the colleagues, I'm reminded every single day just how special this place is. And it's certainly going to be driven home tonight by our featured speaker, Professor Sohini Ramachandran. And given the very enthusiastic response, as you can see from the room to this invitation for tonight's talk, I would surmise that many of you actually share this sentiment, that it's wonderful to have great colleagues like Sohini and so many of you, and people are eager to sort of hear from one another and learn from one another and actually better understand what's going on campus. For me, Sohini is a great example of what we're trying to do here at Brown, showcasing some of the exceptional work and scholarship that takes place here, and in the process, trying to promote a more cohesive community. In my job, I see a lot of conflict in this on this university campus. Some of it silly, some of it not. But what's really wonderful is that when people get together and talk about their work, about ideas and research and pressing, the envelope, then people actually come together and realize why it's so important that we're here together as a community. And it's really wonderful to have you all here. And what's also wonderful tonight is to see several past speakers of these series. So it's great. I mean, every single one that we've done, whether it's been in the evening, whether it's been at lunchtime, they've been packed. And we've also been doing it the last 2 years. We've been having these faculty in focus talks with the staff. So faculty share their research with the staff. And this room is even more packed than it is tonight. So it's really great that people working in a university want to actually learn about what others or their colleagues are doing. So it's really, really terrific. Let me just say a few brief remarks about Sohini. Sohini joined the Brown faculty in 2010. And today she serves as an associate professor of biology, an associate professor of computer science, and she directs the Center for Computational Molecular Biology. And I think what's so exciting, I had the privilege of reading her file as she was being promoted, and I'm not a biologist, I was a pre-med many, many years ago, and the world is a better place that I didn't pursue that career. What is really great about the research is that it's truly interdisciplinary, and it addresses really important problems, problems in population genetics and evolutionary theory, using humans as a study system, and of course employing mathematical modeling and applied statistics and computer simulations, but really asking very important and central questions like, does genetic variation account for different cancer treatments and outcomes? This is super important. Or do cultural traits mutate more quickly than genes? And I had the privilege of once hearing Sohini talk about her research to a group of members of the corporation, and I was riveted. I just thought it was so cool what she was doing to basically broaden the applicability of this kind of work to lots of different populations and not just majority white populations. Now, if you search the Brown website for Sohini, you will find a wealth of articles and news releases about her work, including announcements of critical research discoveries, competitive funding awards, and multiple honors and recognitions that she's earned. For example, in 2012, she was named an Alfred P. Sloan Research Fellow and a Pew Scholar. In 2015, she received an NSF Career Award, and in 2016, she received an NIH R01 for her lab's research on localizing genomic effects underlying adaptive evolution. This year, She was a Natural Sciences Program Fellow at the Swedish Collegium for Advanced Study in Uppsala, Sweden, and was awarded the Presidential Early Career Award in Science and Engineering, which is really one of the very top awards out there. Sohini earned her PhD and bachelor's degree at Stanford, and as noted in the small little biographies on your table, she reports that she was drawn to Brown due to the university's commitment to mentoring young researchers. And I think that's true. I think that when we're at our best, we're not always there, but when most of the time we are there, when we are at our best, we're not only great with our students, we're not only great with one another, but we're actually really great at sort of mentoring younger scholars. And I think Sohini has not only was attracted to Brown, but she's actually embodied that. It's very evident if you look at her lab's website, which is has all these images, really great images of like happy and engaged students, both in the lab and over dinner, and you know, all sorts of blog posts championing her accomplishments, et cetera. In sum, you know, Sohini is everything we love about Brown: an exceptional scholar, dedicated teacher, a real mentor, someone doing important work that's actually going to change the future of healthcare and not just science. And it's truly, I see, for me, a blessing to have Sohini as a colleague. So please join me in welcoming Professor Sohini Ramachandran.
Thank you, Rick, for this wonderful opportunity, and thank you to each of you. I'm incredibly honored that so many colleagues would want to take an evening and sit and listen to me talk, so I'm really thankful for that. And again, that, I think, embodies the wonderful community and connection that we all have at Brown. So I wanted to start with a figure that, for me, embodies my scientific career. And if you're a biologist, you've already seen this figure about 20 times this year. But if you're not, let me tell you about it. So this is the cost of sequencing a human genome as published by the National Institutes of Health from the draft sequence of the human genome that was released in 2001 up till recently. And I like to cast this into relief for myself by overlaying onto it the years that I started graduate school. So when a human genome cost $61.4 million to sequence, to when I started my postdoc, to when I came to Brown, and then when I put my tenure packet in and I stopped updating this figure. So it's obviously just gotten cheaper since then. But this means that for anybody in my field or anyone reading about my field, in the paper, you know that human genetics is no longer data limited, has not been for a long time. But in fact, what we're limited by is the theoretical models that we have to understand these emerging data sets that we deal with. And many people have said this, have said it more eloquently than I, but actually this was the sentence that I opened my postdoctoral research statements with when I was applying for fellowships finishing graduate school. And 13 years ago, I couldn't have imagined the kinds of data sets that we work with routinely in my group today. So this has felt true for a long time, but it's kind of mind-boggling, the routine data sets that we're dealing with that I'll tell you about later. But I would argue that the promise of these data has not been realized, and that we're quite far from realizing it. So what exactly was the promise of these data when the draft sequence of the human genome was published? Well, I'd like to return to this article, which was actually published 10 years later by the National Human Genome Research Institute. So this was the institute at the NIH that Francis Collins directed before he became our National Institutes of Health director. And this was a 10-year plan for 2011 to 2021 that was published in Nature. And I like the title first because it's called Charting a Course for Genomic Medicine from Base Pairs to Bedside. And often in biology, we talk about working from bench to bedside, but I like this phrase because I think it's very inclusive of the importance of bioinformatics and computational biology in the pursuit of personalized medicine. But this is the central figure from this paper. I know those of you in the back aren't going to be able to see it, but I'm just going to point out a few things. First, under different time periods following the planning of the Human Genome Project, these are hypothetical densities of research efforts. So this says beyond 2020. So the idea is we're sort of here, which means that we're really focusing on advancing the science of medicine and improving the effectiveness of healthcare. So this is the ultimate goal. This is a person seeing, I think, a clinician who has a tablet with a genomic sequence on it. Okay? So the idea being that how this person is getting diagnosed, the kinds of traits or diseases that we want to prevent in this person, and the way they're being treated is going to be personalized by their own genome. And I could give you an optimistic talk about how there are many clinically actionable mutations that are being used all the time, including in our own medical school, to treat patients. But today I want to give you the opportunists version, that there's still a lot of work to do if this is our goal. And in fact, what I would argue is that in order to understand the causes and the consequences of genomic variation of any organism, but especially of humans, we increasingly need all of you. Sorry, my battery's run out. I apologize, my computer's a little bit stuck. Let's try again. Okay, there we go. All right, so at the Center for Computational Molecular Biology, we have always been lucky to sit at the intersection of the amazing quantitative sciences that we have at Brown on our main campus and at the School of Public Health, as well as be a critical part of the life sciences arm of our Division of Biology and Medicine. I think for CCMB moving forward in the next 3 to 5 years, though, my vision is that we would draw in more collaborations with our colleagues across campus, many of whom are in this room, who think about what it means to be human, what it means to live in a human society, what it means to provide health care in a human society, and what selection pressures organisms that we rely on and pathogens that we battle are going to face in a rapidly changing climate. So routinely now, I would say that I am thinking more and more about questions that I cannot answer alone and I should not answer alone. And what I'd like to do tonight is to start by talking on behalf of my faculty with you about some of the questions that people who work with genetic data are thinking about today. And I'll just say as a disclaimer that I've biased this to questions that relate more to my work, but I think there are lots of exciting collaborative opportunities with CCMB for many of you in the audience. Okay, so one wonderful partnership that we engaged in the last couple of years was with our Joukowsky Institute here, and I'm very thankful to Peter Van Domelen for reaching out to me, particularly surrounding this wonderful postdoctoral fellow, Dr. Kate Brunson, who was a Joukowsky Fellow. And in order to extend her fellowship, Peter reached out to me, and she is actually going to be joining the tenure track faculty at Wesleyan in just a few weeks. We're going to miss her a lot. But we worked jointly on her appointment for the last year, and she had what I think is the most amazing title for any academic, Visiting Assistant Professor in Biomolecular Anthropology and Archaeology. She basically is Indiana Jones with an evolutionary bent. But what Kate did as a conduit between CCMB and the Joukowsky Institute is she spearheaded last year's State of the Field conference in the Joukowsky, which drew a wonderful set of international researchers from a range of disciplines. And the focus was on the ancient DNA revolution in archaeology. And this was in February. Kate is now finishing a postdoctoral research associate position, being advised by myself, but also by my wonderful colleague, Emilia Huerta Sanchez, who's an assistant professor in ecology and evolution, and also in CCMB. And Emilia is a star in the area of methods development to study ancient DNA, which is very low coverage because it degrades once an organism has died. However, the interdisciplinary problem here is that getting DNA from bones requires very close collaboration between geneticists and anthropologists and archaeologists. it requires taking specimens with consent from and regard for the communities that those specimens are being taken from, the communities that those specimens rightfully belong to. And I would say that Amelia and Kate are real stars at how to do this well, but not every geneticist engages with this in the most appropriate way. But moving to other questions, I think we have some colleagues from anthropology who are in the audience. And could you imagine a world where the person that you date is determined by genetic data? What would be the cultural and social consequences of using genetic data in matchmaking? So it turns out that many communities that practice endogamous marriage already do this. They sometimes do it just through health histories, but now increasingly with actual genetic data. And one example of a company that's been around for over 10 years is called Dor Yeshorim. It operates for Jewish communities throughout the world, out of Brooklyn, but also in Israel. And classical genetics suggests that endogamous marriage over time, inbreeding within a cultural society, is very deleterious, although the results I present later on in this talk will challenge this a little bit. But what happens in Dor Yeshorim testing is it occurs freely in community centers or in schools. And then if two individuals decide that they might want to court each other, they can put their pins into a website and are told the chance that they might have a child with a certain set of disorders. This has led to the severe reduction of Tay-Sachs disease, which is a very debilitating disease that most young children only live a few years with. But it's also led to a change in the autonomy of courtship, some pressures to undergo genetic testing. And one question might be, what is it like for a person who decides not to follow this tradition in their community? The official name of this entity is the Committee for the Prevention of Jewish Genetic Diseases. Building off of that, for economists or political scientists in the room, does enabling prenatal genetic testing alter the kinds of support and infrastructure the government offers to certain parts of society? So if you want to find a beautiful set of articles to read about DNA and our genomes and societal implications, I highly recommend Amy Harmon's series, The DNA Age, which is from 2007. She won a Pulitzer Prize for this series, and it's just absolutely gorgeous science writing. This is one of my favorite articles from it. So it talks about how prenatal testing for Down syndrome spawned the field of genetic counseling. But also that what's happened as a result is that in the US, upwards of 70% of fetuses that are known to have Down syndrome will be aborted. That proportion reaches over 90% in parts of Europe. And what that means is that, as Amy writes in this article, families that have a member that has Down syndrome see less money being put into research on Down syndrome, see less educational infrastructure, and less vocational training. And in the US, this is a population that's quite large, and in fact, that has a fairly long life expectancy, living well into middle age and often being able to live with a fair amount of independence. So what happens when certain, say, states in the United States reimburse individuals for genetic testing? I would argue that prenatal testing is something everyone should have access to, but if then that means that programs are cut because of a bottom line to support certain communities, how should geneticists advocate for that, or advocate against that? So I will say that demographers and geneticists have often written about the consequences of sex-selective abortion in India and China based on one-child laws or family planning in those societies. But we haven't turned inward into our own Western societies to think about this. And just recently, a member of the Icelandic government said publicly, "We have just eradicated Down syndrome from Iceland." So in the face of those kinds of comments, what can geneticists do to push back against that kind of idea? Those of you who know me know that historians are really special to me. I married a historian. And something I think a lot about is our genome's historical documents. So often human geneticists will talk about ourselves as deep historians. But in fact, can a set of genomes tell us about local change in time and place quite recently? How can genomes augment historical studies? I'm going to tell you about an example that has to do with a process that's sometimes called the Great Migration of African Americans from the US South northward. The time period is around 1916 to about the early 1970s. So this is an image from a set of, I think, 57 panels by Jacob Lawrence that's depicting African-American individuals in the South getting on bus lines and heading out of the South to the North. So this study used genome-wide data from over 3,500 self-identified US African Americans and over 9,000 self-identified European Americans. This is a study that was done by researchers at McGill, led by a scientist named Simone Gravel, who's sort of from my cohort in my field. And what they did is they identified genomic segments of relatedness that were generated within the last 5 generations. So a feature of genetic inheritance is that shared sequences between individuals are inversely long compared to how their length is inversely related to how long it's been since those two people had a common ancestor. So the longer stretches of DNA a pair of individuals share, the more recently they're related to each other. Relatedness on this time scale corresponds to shared sequences of about 18 million base pairs, 18 megabases of DNA, and our genome is 3 billion base pairs long. So now I'm going to show you a couple of graphs. So in the self-identified European Americans who are taken from different geographic regions, and this is summarized kind of coarsely, the data were actually quite fine. Here you can see that the edges on the graph are connecting individuals based on sharing these kinds of 18 megabase long sequences in their genome together. So individuals who identify as European American in this data set are connected to people all over. Sort of all pairs of collection sites are connected. There's actually more connection from people in the South with people in the West than in people in the Northeast. But what your eye is probably drawn to is that there's this very strong connection amongst people living in the South. So this means that self-identified European Americans who live in the South today have had family in the South for a very long time, maybe moving amongst geographic locations. But the picture for the self-identified African Americans is different. You see these connections from the South to the geographic North. And in fact, not that strong connected component in the South. And if you look at the more fine geographic data, genetic relatedness often recapitulates, in fact, bus lines towards Milwaukee, towards Chicago, towards Albany, towards St. Louis and Minneapolis that people were taking when leaving the American South. Some of you work on corporate responsibility in the audience. What happens if an employer can discriminate legally against an employee because they have a genetic risk for a disease? So in fact, we of all industrial countries live in the society that has the least protections for genetic mutations that may predispose us to certain diseases. There are differential protections for individuals in the military versus civilians. There's no protection if an employer only employs fewer than 15 individuals. But if the Affordable Care Act is dismantled without the extension of the Genetic Information Non-Discrimination Act, or GINI, then we're all in trouble because the ACA actually extended the protections of GINA to cover pre-existing conditions. And it's been interpreted that genetic mutations are considered, or genetic predisposition is part of a pre-existing condition. So you might tell me, well, I know about the Genetic Information Non-Discrimination Act, and so everything's fine. But what if you make it easy for your employer to get access to your genetic data? So how many people in this room have done a 23andMe or Ancestry DNA kit? Yeah, okay. If you uploaded your DNA, or how many of you have given a kit to somebody? All right, it's like the same people. If you upload that data to a third-party website, that is easily hackable, it's highly susceptible to being able to get your data back, because often when people do that, they're looking for relatives, right? And you can even upload genetic data to some of these websites. You can upload fake genetic data and get back email addresses and names and other measures of genetic relatedness. And this has been used in the last couple of years by police departments in order to solve cold cases. The Golden State Killer was the first such case. And in fact, genetic studies show that this was not luck by which they found these matches. It was actually quite easy to do. So how can geneticists help with these kinds of problems? A question that I've been thinking a lot about, that Rick alluded to in his kind introduction, is from genetic studies of disease in the last 15 years, are we actually just exacerbating health disparities in our own country? Because we don't have equal access to care, and because the diagnostic process that we train doctors with usually involves race as part of the diagnostic process. Well, what are we really learning about genetic predispositions to disease for individuals of different ancestries? This is an image of the cumulative representation of different ancestries in the last 15 years in the International Genome-Wide Association Study Catalog. What you need to know is the red are individuals of European ancestry. And what's shown on the right as a bar chart is, so this cumulative distribution is the proportion of individuals being analyzed in genetic studies of disease of a certain type who are of European American ancestry, or European ancestry. And what you see on the right is the proportion of the world's population that these people actually represent. Okay? So in over 4,000 studies that are called GWASs, or Genome-Wide Association studies, over 90% of the samples only represent 16% of the world's population, and increasingly, less and less of the US population. So what does this mean? What we've learned from this last 15 years of research, is it relevant to all of us in this room or not? This is a question that is what my research program is thinking a lot about these days, and I'm thinking about it closely with another young star at CCMB, Lauren Crawford, who's an assistant professor whose tenure home is in biostatistics. Okay, so now that I've started by telling you what keeps me up at night, what I want to say is that I'm very optimistic, actually, because while I think facilitating true discourse across fields is incredibly difficult, I do think it's something that we're well positioned to do at Brown in a way that my peers at other institutions and computational biology efforts struggle with and aren't able to resolve. And so I really think that this is something that we can do, and we can build out more partnerships in CCMB across the university. And so now that I've told you about kind of the stuff of my nightmares, I'd like to show you the magic of genomic data and the things that we can learn from working in a data-driven way with genomics through a recent project with my group that I'm privileged to lead. So Rick mentioned that these are the kinds of pictures that show up on my website. I will say I recently had a colleague tell me, you know, it's sort of odd. This is a person at another institution. He said, none of the pictures of you in your lab have you in the middle, so I never know where you are. And I sort of said, well, that is kind of on purpose. But anyway, this is the group that I work with, and we're funded by the NIH and NSF and other private sources. So where this project began for me is with a fundamental truism that we all know, which is that we all inherit 50% of our genome from our biological parents. So here are my own genetic experiments. And I was not pre-med either. And so this is kind of the closest to practical biology that I've gotten. But if you had my daughter's genetic data, so they would have one copy of each chromosome from myself and from their biological father, but with very little genetic data and some kind of reference for ancestry, it would actually be quite easy to determine which of their chromosomes they got from their European ancestry parent, their father, and which of their chromosomes they got from me. Okay. And in fact, if you think about it, all of us contain information in our genomes today from about 250 of our genetic ancestors. So I like, if you read poetry, I suggest reading Song of Myself and thinking about this. When Whitman says, I am large, I contain multitudes, this is true for all of us. Right? But when I think about the future of my daughters, if they end up having their own biological children, those children are going to inherit mixed ancestry chromosomes because of a process of meiotic recombination. So what might those chromosomes look like? Well, this meiotic recombination is a fundamental cellular process often studied by cell biologists, and it's a sort of beautiful mystery, but also thought to be a very highly constrained process in most of the tree of life, but not completely. So what happens is, and I'll detail this in a little bit, chromosomes sort of line up in the production of gametes, and they kind of find their buddy and they exchange genetic information. And it's thought that part of this is to ensure that the right number of chromosomes result in the cells that go on to create embryos. So what these chromosomes might look like is they may have 50% of both the parents' ancestries, or 20% of one, 80% of the other. In fact, when we model this, statistically, we model recombination as a Poisson process, a random process in space along the chromosome. Okay, so this is what I've always known to be true and what I've always told my children. But in fact, this project began through a sort of strange post that a customer from 23andMe made on Reddit, actually. So this is a collaboration with 23andMe. And those of you who've done direct-to-consumer testing have probably gotten genetic relatedness results. So what happens in 23andMe, for example, is that if you connect your profile to someone else in the data set, but especially to a parent, you get to see a map of genomic segments that you share with that person through a process called identity by descent. The computational problem of identifying these IBD segments involves some sequence similarity measures and also a hidden Markov model. But this is what a customer posted on Reddit. So here I'm going to show you these are schematics of part of their genome, chromosome 11, 12, 13, 14, 15. And this individual is half identical to this individual's father on all of these chromosomes, as they should be. But then on chromosome 16, there's another picture, this gray color, which I know not everybody can see. But what that is coded as is you are not identical to your father on this chromosome. In fact, this individual has no genetic representation of their father on this chromosome. So this project formed based on this. So we all learned that we get our genome half from our biological mother, half from our biological father. But now I'm going to tell you about people for whom this is not the case. And these people have something called uniparental disomy. The work that I'm going to tell you about just came out last month in the American Journal of Human Genetics. I was co-senior author along with a colleague, Sathira Pangsasuti at 23andMe Incorporated. If people want to ask questions later, I can talk about what it's like to collaborate with this kind of company. It's complicated. But this was led by one of my graduate students who just finished last year in the computational biology doctoral program here at Brown. And here is Priya actually giving a plenary address to 6,000 people on preliminary results from this study at the American Society of Human Genetics. So to tell you a little bit about the data set, 23andMe has over 5 million customers in its database. Ancestry DNA, if any of you have done that kit, has 15 million. They have a lot more. But about 80% of these customers consent to be part of research studies, like the one that I helped lead. And all phenotypic data or trait-based data in 23andMe is self-reported. It's just done through surveys. So what is uniparental disomy? It's the inheritance of both copies of a chromosome from one parent with no representation from the other. So how does this happen? I'm going to first show you what normally happens when we produce our gametes and an embryo is formed. So what normally happens is in this process of meiosis, first homologous chromosomes, they line up, they find their partner, they swap genetic information, and then separate. And then there's another separation of these sister chromatids, and these haploid gametes are formed, like an egg that has 23 haploid chromosomes, meets a sperm, and a disomic zygote. Normal embryogenesis proceeds. In a UPD mechanism, there was a mistake that happened in part of this process. We call it meiotic non-disjunction. If it happened at this stage, we might end up with some gametes that have actually the full complement of chromosomes. So they've got 23 pairs of chromosomes. And then what forms is a trisomic zygote. So this happens in individuals with Down syndrome, some form of this, right? They have 3 copies of chromosome 21. But sometimes there's a process by which one of these 3 copies of the chromosome sort of gets kicked out. And this backup mechanism or trisomic rescue is sort of a holy grail kind of question in biology that lots of people work on. It's hard to know exactly how this happens, and there's not a lot of evidence for it happening in humans. So what's known about UPD to date has come from clinical studies. Individuals present with symptoms in a clinic. They're then referred to genotyping. Usually they're children. Their parents are also genotyped along with them. And the first clinical case was identified in 1987, and there are now about, actually, I think a few more, maybe let's say 3,500 clinical cases of UPD that have been sort of individually reported in the literature. So there's a data set maintained in Jena, Germany, by a physician named Tom Lear. So this is the per chromosome distribution of observed UPD instances in clinical case reports. And what I'm showing here in colors is the parent of origin of the uniparental disomy. So sometimes people get full representation of their father or paternal UPD, and sometimes their mother. But based on these data, maternal UPD is 1.5 times more common than paternal UPD. And many cases are observed on chromosomes where deleterious disorders are produced. And some of these are called imprinting disorders, where the parent of origin of the genetic material is important. This enrichment at chromosome 15 is because there are 2 imprinting disorders that occur based on parent of origin effects on that chromosome, Angelman syndrome and Prader-Willi syndrome. But there are lots of open questions about any rare disease, especially something, or any rare thing genetically, especially something like UPD. So some of these questions might be, what is the prevalence of UPD in a sort of general sample in the population? What are rates of maternal UPD versus paternal UPD or different subtypes of UPD? And what is the etiology of UPD? How does it even happen? Is there a backup mechanism? How do you end up with this occurring? And I should say, an individual can have UPD of multiple chromosomes, and there are actually 12 cases to date of individuals who have genome-wide UPD. So they are not clones of one of their parents. They, in fact, only have genetic material from one parent. They, unfortunately, are not doing so well, but many of them have cancer, actually. So UPD is a very rare phenomenon, and we need really large sample sizes to understand it, characterize it, and actually to reliably estimate its prevalence. Okay, so now I'm going to tell you about 3 subtypes of UPD, and then I'll move on to computational questions. So depending on when the error in chromosome separation happens, you can actually end up with something called heterodisomy, where one inherits both copies of a chromosome from one parent. So represents 2 grandparents, but just their maternal grandparents, not all 4 grandparents. So this is called heterodisomy. If non-disjunction happens later on, you can end up with what's called isodisomy. And the reason why I have a couple of stars here is isodisomy means complete identity of your genetic data across an entire chromosome. This is thought to be incredibly deleterious. Genetic variation is important. If there's a deleterious mutation that someone inherited from this parent, they would have 2 copies of it. This is one of the reasons we dissuade people from breeding with close relatives. The idea is that eventually you would produce this kind of pattern. So this is classically thought to be incredibly deleterious. And then another form is where crossing over has happened, but there's still some isodisomy, and I'm going to call this partial isodisomy. But again, this can still produce sequence identity along, say, 1/3 of a chromosome, so incredibly long stretches of DNA. So I'm going to use these different cartoons to represent these different forms of uniparental disomy. But now I'm going to tell you why a computational molecular biologist might be interested in this sort of interesting cell biology question. And the reason is that we can actually use the different patterns that we predict to occur when UPD happens to detect UPD computationally in large data sets like the 23andMe database. So I've told you about different ways that UPD forms and the different subtypes of UPD, but now I'll tell you how we can detect it. We can do it 2 ways. One is to use what I'm going to call runs of homozygosity, and all that means is just long runs of no genetic difference between both pairs of a chromosome in a single individual genome. In a case of heterodisomy, we actually don't expect to see any long blocks of homozygosity because this individual has inherited all the genetic variation just one of their parents had of this chromosome. But in isodisomy and partial isodisomy, we can use these long blocks of a lack of genetic variation to detect UPD. If we have parent-child data, we can actually identify true positives and true negatives using patterns of identity by descent. So what 23andMe or Ancestry is just reporting to individuals, on a UPD chromosome, a child will not share any genetic material from one of their parents. Okay, so if we look for UPD in 23andMe parent-child pairs, which turns out to be a very rich data set because they do pushes every Mother and Father's Day, and everyone likes to buy their parents kits, then we actually find across 1 million parent-child pair duos, which includes some trios, a child and both of their parents, we find 199 true positives for UPD. We see cases on almost every chromosome, except chromosome 18. We're able to identify the parent of origin as long as we have one of their biological parents. And I'll say that all of this is based on genetic relatedness findings. So we aren't using, we're finding the biological relatives ourselves, not using a parent someone has identified as connected to them in the data set. So from these distributions, what we see is that maternal UPD is in fact 3 times more common than paternal UPD, which is quite different from what the clinical data show. And we see many cases on a different set of chromosomes than we saw in the clinical data. Notably, this mode is at chromosome 16, not chromosome 15. The existing estimates of prevalence from clinical data, which I would argue were not really estimated in a very good way, are between 1 in 3,500 and 1 in 5,000 individuals having UPD. And our estimates are more than double that, 1 in 2,000 individuals using this data set and only using trios. So it turns out that the cases that we're observing are quite different from what we see in the clinical data set. Those cases are biased towards chromosomes where an individual then presents with symptoms in a clinic and gets genotyped for diagnostic purposes. So what I'm going to show you here are, on the y-axis, the per chromosome distribution I just showed you on the previous slide, and on the x-axis, the per chromosome distribution from the UPD database of clinical case reports. And here I'm coloring these based on the type of centromere these chromosomes have, which might influence their stability in cell division. But what I want to say here is that in genomics, even with 23 points, it's really hard to get a correlation that statistically is 0. And that's what's happened here. So I can't reject the hypothesis that this correlation is 0. In fact, so these data sets are telling me about very, very different things. Also, initially when I saw these results, I thought, oh, we're seeing children who have UPD and their parents, went to CVS and got a 23andMe kit instead of going to a genetic counselor. It turns out that the median age of the UPD true positives we identify is the median age of customers in the data set, which is over 50. So these are not young people. So the computational question that's actually interesting is not what I've told you about. It's how do we detect UPD for an individual without using any parent data or any parent-child data? So the way that we're going to do this is to use a test of homozygosity. So we're just going to look for long stretches of DNA with no variation within individual genomes. So now I'm going to show you some pictures that are karyograms. And so they're just schematics of the 22 non-sex chromosomes. And on those, I'm going to color a really long block of homozygosity that's more than 1 million base pairs long as green. So even if you're in the front, you're not going to see anything here because this is a true negative. This individual does not have UPD. They have very few of those blocks, only about 4 of them scattered throughout their genome. Here is a true positive for heterodisomy of chromosome 1. So this individual also does not have long blocks of homozygosity because, again, they've inherited both copies of one of their parents' chromosome 1. But here's an individual with partial iso-UPD. So even if you're in the back, I think you can see these long, long blocks of green on chromosome 1. And here is a true positive for isodisomy of chromosome 1. So again, some of this might be the fact that I was an applied math major in college, but when I first saw this, I thought, how is this happening? Chromosome 1 is the largest chromosome in our genome. It's a huge amount of genetic material, and this person has no genetic variability on this chromosome. Okay, so this sets up a classification problem in a machine learning sense. I want to distinguish the 2 pictures on the left from the 2 pictures on the right. I want to train a computer to do that. And it seems like maybe not a hard problem, right? So how does it become a hard problem? It becomes a hard problem when an individual comes from a community where individuals might be, their parents might be distantly related. And this could happen in any community. It can happen in an Ashkenazi Jewish community, in a South Asian community, like the one I'm from, in a European community. So now I'm going to show you what UPD on chromosome 1 looks like for individuals with distantly related parents. So now there are long blocks of homozygosity on many different chromosomes, not just chromosome 1, which is where the UPD event has happened. Even in the individual with heterodisomy, we now have these long blocks. And now even the control individual has long blocks of homozygosity because of the pedigree that their genome has traversed. So it turns out these runs of homozygosity are going to vary in their length based on chromosome by chance, but also based on ancestry. And now this is the classification problem. How do I train a statistical algorithm to classify these pictures on the left as different from the 4 pictures on the right? So the way that we did this is through extensive simulation and what's called supervised classification. We trained balanced logistic regression classifiers for every chromosome. We did it in every ancestral population that 23andMe identifies. And we used a lot of independent variables for prediction. The reason why we used regression is we wanted to understand the features that were important for the classification. So it turned out that what was very important was features of how runs of homozygosity were distributed across the genome, and particularly how long was the second longest run of homozygosity, because that helped us understand if we were seeing a product of consanguinity or an individual with UPD. And then we had to pick a threshold, and we were very conservative because we had all these true positives that we wanted to classify properly. So when we do this and we look in almost 1.4 million what are called singletons at 23andMe, individuals who do not have a parent in the database, we find 304 putative UPD cases. I now can't color them by parent of origin, but they span every chromosome. This recapitulates features of the per chromosome results I showed you when we used parent-child data, so the distribution is quite similar. We were able to replicate this in another data set called the UK Biobank. The UK Biobank data are genomic data and medical records from the British National Health Service for 500,000 residents of the UK. They also have imaging data now. It's kind of an amazing data set. Because the UK has undergone this effort, now everybody who works in human genetics is working on the genetics of British people. So it's sort of a strange service we're all providing now to the UK. But this analysis was done by a third-year computational biology student, Sam Smith, in my group. And again, this is correlated with the distributions I've already shown you from 23andMe. So we're able to find another set of cases in this other data set and replicate. So where does UPD come from? Well, there are lots of open questions still about it, but one way we tried to get at this is to try to look at other data sets where we knew there had been an error in meiosis, in chromosome segregation. So now I'm still showing you our analysis using parent-child data on the y-axis in 23andMe. And on the x-axis, I'm showing you a published data set of per chromosome aneuploidy rates that are from pre-implantation genetic screening from in vitro fertilization data. So embryos in an IVF clinic are screened for chromosomal abnormalities, and this was a publication of one such set of aneuploidy rates. So now you can see that there's some correlation between these two sets of per chromosome rates. In fact, now what we're trying to pursue is whether this is correlated with UPD that happens in cancer. So there's what's called segmental UPD that can occur in tumors. And we want to see if what we're detecting is just that certain chromosomes are more unstable routinely in cell division than others in all different kinds of contexts. So what have we learned about a fundamental biological question? How does genetic inheritance happen in individuals like us that have two parents from computational work? I think we've learned a lot of things. One is that we've estimated a prevalence that's much higher than previously thought from a more general sample than UPD has been studied in previously. So we actually estimate that this is not that rare a genetic situation people encounter. And what this tells me is that there is strong evidence that there's a backup mechanism that corrects meiotic errors, which really hadn't been established in humans before. That there could be one that could correct meiotic errors on all chromosomes. There's some data for chromosome 22, which is our smallest non-sex chromosome, but not genome-wide. Also, we are detecting UPD in individuals who have not been seen in a clinic because they had a consequence of having UPD, and who in the UK Biobank do not report any UPD consequences that are common imprinting disorders or any kind of aneuploidy diagnosis. So this tells me that while there may be embryonic selection against meiotic non-disjunction, that in fact live-born individuals who have had this error corrected may be deficient for deleterious mutations. And there's some suggestion now in analyses of Down syndrome that this is also true. Increasingly, some evolutionary studies of domesticated animals are also showing that while animals that we sort of force to breathe together in certain lineages do end up having deleterious diseases over time, the burden of deleterious diseases they have is much less than we think they might have. And that's sort of conditional on who survives to be born. So I'd like to close by telling you about how Eric Lander, who's the head of the Broad Institute, Harvard and MIT, and led the government-sponsored sequencing effort for the human genome, this is how he summarized the state of genomics in 2003. So this is when the Human Genome Project sort of officially closed, and he was giving an important lecture, which is the Ig Nobel lecture. He gave the Nano lecture, which is, I think, 24 seconds long, and you have to write a 7-word or less abstract. And he wrote a great one. Genome, bought the book, hard to read. It's a great abstract for genomics in 2003. So I'm not sure what Eric Lander would say today, but I can tell you what I think of as the abstract for this talk, and what I'd like to say to you. Genome, incredible book. Let's read it together. Thank you.
We have about 15 minutes for questions. So
Yeah.
Let me feel it.
Yes, I feel this.
Thank you. This is great. I was wondering whether the differences in the frequency of uniparental disomy between the cohort that had the sort of detected by illness population versus the casual cohort through 23andMe really demonstrates that perhaps some of the chromosomes that have high incidence in the disease population are more prone to leading to disease consequences. So the difference between 15 and 16 may reflect that, perhaps? Or?
Yeah, so I think most of those cases are enriched for imprinting disorders. And so that's where complete homozygosity will be an issue. So yes, I think definitely it's that, sorry? Defining an imprinting disorder. So an imprinting disorder is when the function of a gene depends on the parent that it originated from. So there's a sort of memory that can happen where the paternal allele gets silenced or upregulated in the individual at different stages of development. And in fact, this kind of genomic conflict is sort of crucial to gestation. So parental alleles that promote growth in utero are usually silenced so that gestation can happen properly and then kind of upregulated later on in development. So yeah, I think you're absolutely right. It's that what's known about UPD has come from situations where UPD had a clinical consequence. I think what's interesting about this study is to think that, if we reach back to our high school biology, we sort of learn that genetic variation is good, which is true, and that, but that a lack of genetic variation is bad. And here are individuals for whom that might not be the case. And we may never see them or know about these errors in cell division without looking in a different kind of sample. Yes, Ken.
I'm really interested in the process of trisomy rescue. Mm-hmm. And I wondered, when I looked at the differences between different chromosomes, if the fact that some chromosomes are strongly represented in terms of the disomy statistics and others are not, is that because trisomy occurs more frequently in those chromosomes, or because trisomy rescue is less effective in those chromosomes?
Yes, that is the question.
Just ask it.
Yeah, so I don't know, and that's not detectable from these data. So the data that people, even in population genetics who study recombination, sort of hope for is access to pre-implantation diagnostic data to ask that exact question, where you could sort of see aneuploidies happening and then figure out which ones kind of got rescued or not. But I do not have access to such data. But I think that's a great question, right? So are we seeing situations where an error has... Yeah, it would be a mistake to infer from these per chromosome distributions that these are the chromosomes on which, these are the only chromosomes on which trisomy is happening or trisomy rescue is happening. I think you're right. So I can't answer that question, but I agree with you that that's the question in a lot of ways. Yes?
I'd love to invite you to speculate on something you didn't actually talk about, and that is a recent article in the New York Times commented that the Chinese, who have incredible sequencing resources, were trying to correlate genome with facial morphology. What would you speculate their chances of coming up with a reasonable R value would be?
Well, with enough data, you can always get a correlation. A lot of, in statistics, there is always a big discussion about is a p-value, what's it worth? I would say, what's a correlation coefficient worth? But that is a great question. So in fact, that has already happened in the US. So, Mark Schreiber, who works at Penn State, has identified a number of mutations that are useful for determining facial morphology, and these are also being used to solve cases, to sort of generate images like what a sketch artist would make. So I would say I think that they actually do have a chance of doing that. The motivation is, I don't know why, but I've heard Mark Schreiber talk about this a lot, and I know he thought about it very thoughtfully, and he's an anthropologist and a geneticist, and he embarked on a really long study that's quite interesting about how we can easily, right, classify individuals and their ancestry just by looking at each other, but actually trying to understand what in the genome creates our facial structure is quite complicated. But he has identified some large effect mutations that can predict certain features of the face. And that work came out a few years ago. But I would say maybe look at that if you're interested in that question. Yes.
Did you identify any cases in the 23andMe data, either from trios or duos, where the parent also had UPD? And I'm just thinking if there's a way to identify a mechanism that's driving the prevalence of UPD within family.
Yeah, that's a great question. So we were underpowered to ask that question. And I will say we didn't, well, I actually don't know. I think we looked, what I want to say is that we didn't see any individuals with UPD who reported having children in 23andMe, but not everybody fills out survey data, and it's not totally clear to me whether that's the case or not. There has been a recent study out of David Wilson's group that was published in Nature Communications just in the last month on autozygosity or homozygosity and various phenotypes associated with it, and they show some suggestive data that individuals who mate, who both have sort of long runs of homozygosity have fewer children. But it's still unknown whether that's kind of a biological consequence of the autozygosity or just correlative. So yeah, I don't know the answer to that question. Yeah, David.
Is there, from the parent data, is there any evidence that the incidence of UPD correlates with individuals who are more closely or distantly related as parents?
That
sort of
is,
back to the first part of your talk.
yeah, so
I mean,
that is a good question. We did those I'm not sure that we tested that exact question, but we did see an association with maternal age, but not with paternal age. But we weren't able, so again, going back to kind of the data everyone wants, the pre-implantation diagnostic data to study recombination, the ideal thing to do there would be to take the parents' genomes and do an association study between, is there something in a parent's genome that induces an error in meiotic non-disjunction? But again, no one's done that data. We were underpowered to do that. So, yeah, Chris.
Yep. So I'm just a dumb engineer, information theorist, no biology. But is the information, is all the information in the genome, or and I'm kind of hinting at epigenetics too, but I'm also hinting at, I'm pretty sure there were studies where you can pull a set of chromosomes out, or a fertilized, I don't even know what to call it, but the genetic information, reimplant it in another egg, and it develops differently, but it still develops. So is all the information, it seems like there may be things that we're missing, even with 3.2 billion base pairs?
Perhaps, yes. So that may get to the previous question about kind of consequences of UPD down the road. Yes, that could be true, and I would agree with you that there may be some non-genetic factors at work. And I still feel comfortable with what I presented being kind of a genetic process. So
I'm just looking for something else to apply my trade.
yeah. Well, no, but it is a fascinating thing. I mean, even with pre-implantation diagnostic testing, it's removing, a couple of cells at a very early developmental stage, and the embryo still develops normally. And in fact, you can separate and have two embryos. I mean, it's like kind of amazing. So,
But the 23andMe data does not contain any.
it does not contain any epigenetic data. No, that is
We
yes.
have time for one more question.
Okay, well, you're the last one then.
Thanks. Yeah, thanks for the great talk. I'm curious to know more about the role that, or to what extent machine learning was helpful in your project. Like, you've been talking about all these different questions that you've answered with the data that are really fascinating. Like, you have two versions of the 23andMe data, right? You have the stuff that you're really confident about and the expanded data set that you've identified with machine learning.
Yeah.
Did you look at how these questions might change, like or the answers to these questions might change depending on what data set you were looking at? Could you say that, because we used machine learning, we were able to reach this better conclusion? Or does it just kind of give you the more confidence?
I would say it enables us to do the classification without the parental data, where we are almost totally confident that we're right.
Mm-hmm.
So that's one thing. And we are then able to use those true positives and true negatives as a way to validate our classifier. But I guess what I would say is it really has to do with that problem of, so often in genetic data, the applications of machine learning are really useful, but everything's trained. It's based on sort of supervised classification. And here we have a ton of data, a ton of true negatives we can use to kind of simulate recombination, or true positives that we can use for training. But I would say the challenge that we still are sort of trying to solve is that actually, what I didn't tell you, is that in some of these ancestries, Ashkenazi Jews being one of them, it's still incredibly hard because of how much homozygosity there is in these individuals to actually do the classification. And so there, I think that computational technologies could potentially help us if we had the right, even if we had much more training data or something. But I'm not sure if that totally got at your answer.
Yeah, it was also just, what if you had, some skeptic could say, well, what if you just did your analysis on the data that the true positives and the true negatives that you had?
Mm-hmm.
Would that change the conclusions?
You mean in terms of what? In terms of estimating prevalence?
Yeah, things like that.
Yeah, so those kinds of things we did do only with, so we didn't see differences in estimation of prevalence if we used both data sets. And I guess that I would say that's probably the other thing we were trying to do besides identify particular cases. One thing we did see was a lot more sex chromosome cases of UPD or aneuploidy in the UK Biobank specifically. It looks like there are a lot of individuals who might have a deletion on the X chromosome, which if we had only looked at parent-child data we wouldn't have seen, maybe. So those are a couple of the examples I can think of.
Great, thanks.
But I'd love to talk more about it another time.
Please join me in thanking. Thanks for coming, everyone, and have a great end of the semester.